We live in an age of unprecedented scientific advancement. The ability to edit the very building blocks of life, our genes, is no longer a figment of science fiction, but a tangible reality. At the forefront of this revolution is CRISPR-Cas9, more commonly known as CRISPR, a technology that has taken the scientific community by storm. CRISPR offers a level of precision and ease in gene editing that was previously unimaginable, opening doors to potential cures for genetic diseases, advancements in agriculture, and even raising profound ethical questions about the future of humanity. The excitement surrounding CRISPR is palpable. Scientists around the world are exploring its vast potential, driven by the hope of alleviating human suffering and improving lives. Yet alongside this optimism lies a sense of caution. The power to alter the genetic makeup of organisms, including our own, comes with immense responsibility. The ethical implications of CRISPR are complex and far-reaching. We must carefully consider the potential consequences of our actions, both intended and unintended, before embarking on this uncharted territory. This essay explores the intricacies of CRISPR, delving into its mechanisms, potential benefits, and the ethical concerns that accompany this revolutionary technology. CRISPR-Cas9 is often described as a pair of molecular scissors. This analogy, while simplified, captures the essence of its function, the ability to cut DNA at specific locations. This precision is what makes CRISPR so remarkable. Previous gene editing techniques were cumbersome and prone to errors. CRISPR, in contrast, offers a level of accuracy and efficiency that is revolutionizing the field of genetic engineering. The system consists of two key components: the Cas9 enzyme, which acts as the molecular scissors, and a guide RNA molecule. The guide RNA is a short sequence of genetic material that is designed to match a specific target sequence in the DNA. Think of it as a GPS system that guides the Cas9 enzyme to the precise location on the DNA strand that needs to be edited. Once the Cas9 enzyme, guided by the RNA, reaches its target location, it makes a cut in the DNA strand. This break in the DNA triggers the cell's natural repair mechanisms. Scientists can exploit these repair mechanisms to introduce changes in the genetic code, either disrupting a gene's function or inserting new genetic material. This ability to precisely modify DNA sequences holds immense potential for treating genetic diseases, developing new agricultural crops, and even controlling the spread of infectious diseases. The potential of CRISPR in medicine is immense. Scientists envision a future where genetic diseases, currently deemed incurable, could be treated or even cured by correcting the faulty genes responsible for these conditions. Imagine a world without cystic fibrosis, sickle cell anemia, or Huntington's disease. CRISPR offers a glimmer of hope for millions of individuals and families affected by these debilitating and often fatal conditions. The ability to target and modify specific genes implicated in disease opens up a world of possibilities for developing targeted therapies. In the case of cystic fibrosis, for example, CRISPR could be used to correct the faulty gene responsible for the production of a protein that causes mucus buildup in the lungs and other organs. This correction could potentially prevent or even reverse the debilitating lung damage that characterizes the disease. Similarly, CRISPR holds promise for treating other genetic diseases. Sickle cell anemia, a blood disorder characterized by misshapen red blood cells, results from a single mutation in the gene that produces hemoglobin. CRISPR could potentially correct this mutation, allowing individuals with the disease to produce normal, healthy red blood cells. The implications are far-reaching, potentially freeing patients from a lifetime of pain, complications, and reduced life expectancy. While the potential benefits of CRISPR are undeniable, the technology also raises profound ethical questions. The ability to manipulate the very essence of life, the genetic code, forces us to confront fundamental questions about our values, our responsibilities, and the future we want to create. One of the most pressing ethical concerns is the potential for creating designer babies. CRISPR could theoretically be used to modify embryos to select for desirable traits such as intelligence, athleticism, or physical appearance. This raises concerns about exacerbating existing social inequalities and creating a society where individuals are valued not for their inherent worth, but for their genetically enhanced capabilities. The slippery slope argument, often used in ethical debates surrounding genetic engineering, warns of the potential for unintended consequences. While the intention to eradicate diseases like cystic fibrosis is noble, the line between therapy and enhancement can be blurry. What starts as an effort to alleviate suffering could lead to a future where parents feel pressured to enhance their children's genetic makeup, blurring the lines between natural variation and engineered perfection. The ethical dilemmas surrounding CRISPR are complex and multifaceted. There are no easy answers, and the decisions we make today will have far-reaching consequences for future generations. We must proceed with caution and engage in open and informed public discourse to ensure that this powerful technology is used responsibly and for the benefit of all humankind.